Assalamu alaikum and very good afternoon, ladies and gentlemen. So I'm Dr. Muhammad Usman Rashid, Senior Research Scientist and Head of Basic Sciences Research. So with me, the chair of the session is Professor Dr. Ziaullah Khan. He is the Vice Chancellor and Dean of Public Health, Khyber Medical University, Peshawar. Over to Dr. Ziaullah Khan.
Thank you so much. It's really an honor and pleasure to be here at this very prestigious conference in which we have delegates from different countries. I've been told that all of them didn't come, but due to many reasons. But we have their recorded talks, and we have Professor Talu here, luckily here, so he will be talking in person. So
our first speaker is Professor Rodney Scott. He's a professor of medical genetics, the University of Newcastle, Australia. So he's going to talk about, are there germline genetic variants that correlate with mutational events in adult brain tumors? Professor Rodney Scott.
Good afternoon, everyone. Firstly, before I give my talk, I'd like to thank the organizers of the committee who invited me to give my presentation today. That is Drs. Mohammed Usman Rashid, Dr. Kashif Syed, and Dr. Usman Ahmed. I trust that the 22nd Shaukat Khanum Cancer Symposium is so far, or has so far been a success, and that the organizers are not too stressed about the symposium anymore, and that they're able to relax and listen to the talks without having to worry about organizing the speakers and every other issue that comes up during these sorts of meetings. I'd also like to thank the audience for their participation, be they in person or online, and trust that they gain something useful out of this presentation. So I'll share my screen now and take it from there with respect to what I'm going to talk about. Excuse me a minute. So what I'm going to talk about today is listed in the title, are there germline genetic variants that correlate with mutational events in adult brain tumors? Now, this work couldn't have been done without the help of Drs. Jiju Summer, Zhang, Dr. Andrea Johns, Dr. Alexander Xavier, Dr. Kelly Kalida, without which I wouldn't be here today being able to present this data. I'd also like to put out a big thank you to the Mark Hughes Foundation for their contribution to brain cancer research. Just a little bit of background on the Mark Hughes Foundation. It was founded by an individual who was unfortunate enough to be diagnosed with a brain tumor. Fortunately, it wasn't a high-grade tumor, and he is doing well currently, and has been now 3 years. It has been now 3 years since, I think, 3 or more years actually, since his diagnosis. So he's doing very well. And he was a football player in one of the national leagues here in Australia. And he and his team got together and decided to create the Mark Hughes Foundation for Brain Cancer Research. And it has done exceedingly well and continues to do well on an annual basis when they have their major drives to support brain tumor research. So what are we going to talk about today? Well, we're going to be looking at the constitutional DNA samples from individuals diagnosed with brain tumors, and we're going to compare some of those to, or the majority of them, to 128 matched tumor samples that we were able to obtain. The cohort consists of a variety of brain malignancies, but glioblastoma multiforme is by far the largest contingent, and that's represents around, and I'll show this in the following slide, around 75% of the total number of brain tumors that we have in our bank. The age of the patients was ranged between 17 years to 84 years of diagnosis, the majority of which occurred between the ages of about 55 years of age and 75 years of age, as represented by this diagram here. The brown bars represent glioblastoma multiforme. The blue bars represent other less aggressive brain tumor types. This is again showing this flow diagram, and by far the largest, as I mentioned, were glioblastoma multiforme tumors, but there were a range of other brain tumors that were studied as well in this investigation, and we looked, and they're listed here. I won't read through them, but they are here. There were multiple cases of some of them, and altogether they represented 25% of the cases that we interrogated, which gave us some idea as to whether or not there were specific differences between lower and high grade tumors, and we'll come back to that in a minute. What we did was whole exome sequencing on genomic DNA that was extracted from peripheral blood lymphocytes, and we also did whole exome sequencing on formalin-fixed parental embedded tissues. And we used the Queer amplification kit and the Agilent SureSelect exome version 7 library preparation kit, which allowed us to get over 48 megabytes of coverage per person. And we sequenced these on a Illumina NovaSeq 6000 platform. The analysis of the data was undertaken by GATK, an analysis tool developed by Broad Institute in the US, and the Codex 2 software package that allowed us to interrogate the presence or absence of copy number variants. So, looking primarily at germline changes, what did we observe? Well, first thing we can say is there was no obvious pattern of germline change that we could identify in this cohort. Well, this diagram here represents the top 19 hits that we identified. And some of them are interesting, we'll go into them in a few minutes, but the most important thing to look at when you're looking at this diagram is the number of green rectangles that occur in the diagram. And these are associated with missense mutations, and there's many of those, and by far the majority of them are impossible to interpret with respect to their pathogenicity. And these are probably termed variants of unknown significance, or they're likely benign or benign, and most of them will be benign. However, we did identify some interesting and intriguing hits, even in these 19 genes, that suggested that something different was going on in these genes than just common polymorphisms, or polymorphisms that have not yet so far been reported. So there were a number of nonsense mutations, splice-like mutations, frame shift deletions, frame shift insertions, in-frame insertions and deletions, and multi-hits, represented by the black rectangles. Of particular note is that some genes contained more changes than others, and some of the changes were more likely to be nonsense mutations or in-frame insertions, which may or may not alter the protein product and give rise to a change in risk. If we look at the bottom part of this diagram, we see this orange bar here, and then a multicolored bar here, and that represents the 75% of glioblastomas and the remaining lower grade tumors that we were able to identify. Of particular note, what we observed in this overall diagram is that there is a difference between the glioblastomas and those tumors with lower grade disease. And that's shown here, where we can see that there's a demarcation between the glioblastomas, indicated here on the left, compared to the lower grade tumors that we were able to study. What we couldn't identify was any correlation between the age of diagnosis and survival, number of mutated genes, and germline mutational burden, which suggests in this cohort, and I'm only talking about this cohort, that there wasn't anything specific at the germline level that predict whether or not somebody was likely to present with a brain tumor, or on current knowledge was likely to present with a brain tumor. Some of the germline variants are listed here again, and two of them are quite interesting. MBPF20 which are neuroblastoma breakpoint family of proteins, and IGFN1, which is the immunoglobulin-like and fibronectin type 3 domain containing one gene, that's what it stands for, and it's associated with the immune system. These were also present in a pediatric brain tumor cohort that we identified, and they may signify something. A number of the genes that we identified were intriguing and could be associated with brain tumor development, and these included the innate immune system problems associated with PRSS3, C3DC168, which is associated with adenosquamous carcinoma, and a number of other genes that are listed here that are associated with colorectal cancer or ulcerative colitis, and genes that are associated with microtubule-dependent spindle pole assembly. Suffice to say, there are a number of genes here that don't really relate to anything. The ciliopathy gene, KIA0556, is probably just a gene that we identified that is relatively commonly mutated in the general population. There could well be some other ones as well. So the other aspect that we didn't identify in our cohort was that there were no known germline suspects. That would include genes that were associated with DNA mismatch repair, and this could be linked to Lynch syndrome, where we know that every now and again you see patients with glioblastomas presenting with a microsatellite instability phenotype, which is what you observe in Lynch syndrome. There were no APC mutations, especially not in the lower grade tumors. So any gangliomas that we looked at didn't carry an APC mutation that we could tell. There were no germline TP53 mutations associated with the Lee-Fraumeni syndrome, and that syndrome is linked to increased risk of brain tumors. In addition, the genes that we did identify, especially those top hits that we identified, there wasn't much revealed in the database genome with respect to confirmed genomic changes associated with associated with germline changes and the link to disease development. Some of these genes are likely to be common, as I mentioned before, in the general population, and not likely to be associated with brain tumor risk. But the one question that we actually had a closer look at was were any of these germline changes associated with prognosis? It's one thing having a genetic change that's linked to the risk of developing disease, but some of them may actually be linked to whether or not disease progresses much more rapidly in one group of patients compared to another. We sort of were able to interrogate them, and we did paired variant analysis, and that is you take the pairs and you do a reiterative analysis to check or to determine whether any of them could be linked to poor prognosis. And sure enough, in the top 19, there were two genes that we were able to identify, ADPRHL1 and STARD9, which appeared to be associated with the prognosis of disease. So if you have mutations in ADPRHL1 and STARD9, your prognosis appeared to be much worse than what it would have been had you not had mutations in those two genes. ADPRHL1 is associated with brain small vessel disease, which could theoretically be linked to an increased risk of brain tumor movement away from its initial site into sort of the surrounding tissues. And STARD9, which is a microtubule-dependent motor protein required for spindle pole assembly during mitosis. And if that's mutated, that may, in fact, result in an increased rate of mutations in those cells that don't have appropriate spindle pole assembly. So there are two intriguing factors that we identified that were associated with worse survival in our cohort. So in summary, with respect to the germline single nucleotide variants and insertion deletions that we identified, there's no evident molecular signature for predisposition to brain tumors. None of the known susceptibility genes that have been reported previously were represented in this cohort. The cohort was mostly aged over 40 years of age, suggesting possibly that environmental factors may play a more important role in disease development compared to genetic factors. One gene pair we just mentioned appears to be linked to survival, and it points to the role of epigenetic change, and this is change that occurs on the genome sequence but doesn't change the sequence, that may provide more information on the risks of developing brain tumors, and we're currently interrogating that right now. It's not ready for any sort of public announcement. So, what did we find in the somatic study that we did, looking at the brain tumors themselves? Is there anything there that we could get a handle on? Well, the first thing we did, we actually compared our cohort to the TCGA cohort indicated here by the red arrow, and our own cohort is indicated by the red arrow going in the reverse direction, and it shows that both cohorts are somewhat similar. They both contain tumors that have higher mutational burdens and those that have some lower mutational burdens, but the average burden is about the same, indicating that our cohort is not too different to smaller cohorts that have been studied elsewhere. The mutational profile does seem similar in this diagram to what I showed you before. There's a mix primarily of missense mutations again, but also represented in this cohort are nonsense mutations, splice site mutations, frameshift deletions and frameshift insertions. These are important because they always predict, virtually always predict, a mutated protein product. In-frame insertions, deletions, which may not predict a mutated product, and multi-hit changes in particular genes. Some of the genes will have multi-hits in them because they're large and it doesn't actually affect them too much. And these multi-hits may in fact disguise some of the missense variants that are shown here. Notwithstanding, we were able to pull out some interesting information in that there were a number of tumors within our cohort that displayed hypermutability, and this could be reflected in a DNA repair system, which we may come to in a minute. Some of the somatic single nucleotide variants and insertion deletions have been reported previously. So we did find this time in the tumors the oncogenes that included TP53, PRSS3, and EGFR were mutated coincidentally in this tumor cohort. The usual suspects of brain tumor mutations were also identified, IDH1, P10, and F1. They were also identified and seen in this diagram. And also, intriguingly, as we just found, that PRSS3 also seen the germline was represented here in this cohort. So the cohort is representative of events that occur within brain tumors that we know about, but also pulls out a whole range of other genes that we haven't otherwise noted, and that's listed, as we see in this diagram here. If we look at the top 25 driver genes, and these are genes associated with driving tumorigenesis, we actually see that IDH1 comes out of position 2. It's a key gene that we already knew about, so that tells us that our cohort is in fact representative. A new one, DDX11, came out as the top hit, and we'll have a little bit of a look at that in a second. That was an interesting one, and there's a number of other genes here that are of particular interest and demonstrate that something's going on at the somatic level to cause mutations in these genes which drive brain tumor development. If we look at some of the signaling pathways that have been associated with the genes that we've identified, we see that the top two are the RAS pathway, as in top one, indicated on the left-hand side here, and the Wnt signaling pathway, as indicated in the top two slide over here. And if you look at the y-axis, it's the genes that are involved in these pathways, and X along the y-axis along here, which you can't really see, is the number of samples that had something in them. So clearly EGFR, which comes out as one of the top genes in the RAS pathway, is often affected, and another top gene, NF1, is also affected most often. Whereas in the top two, when you're looking at the Wnt signaling pathway, there are a number of genes that are associated with Wnt signaling there. And one of the ones that's actually quite intriguing is AXIN1, which is also clearly implicated in the APC pathway and is one of the top hits that we identified in the Wnt signaling pathway. We could show you more information looking at the Notch pathway, the Hippo pathway, the PI3 kinase pathway, etc. And these are important pathways that need further interrogation, which we don't really have time to go into today. If we look at survival and some of these changes, we see that P10 is a key driver, and if you have a mutated P10, your likelihood of survival is much, much less compared to those individuals who don't have a P10 mutation. Similarly, for ADAM29, that's another gene that we identified. There was a significant decrease in survival if you had a mutation in that gene. And DDX1, which is our top hit, also is associated with poorer survival if it had a mutation in it. So this is some information that we can look at. It's prognostic, potentially, and it could be quite valuable. You can combine this, and we looked at 3 mutations, somatic SMBs again, and indels, and compared those to survival. So we had MBPF1, which was in an earlier diagram showed that this was one of the major pathways that was hit, or major gene families that was hit in brain tumors, and compared it with DDX11, and we found clearly that was an association linked with poorer survival. MBPF1, MUC12, and MAP2K3 is also associated with poorer survival if you have mutations in those 3 genes, and MBPF10 and MUC3A and KCNJ12. If you have mutations in those 3 genes, you also see poorer survival. Now, these are the ones that we identified in our top hits. There are undoubtedly more, but it does tell you that we are able to provide prognostic information based on this sort of information. Whether it is of any benefit for the patients or not is open to question, but it does begin to open the door with respect to identifying potentially new targets that may reduce the relapse rates in patients, unfortunately, to be diagnosed with these tumors. If we looked at the mutational signature of these tumors, we found that they're identical to what has been identified before, in such that they're 4 signatures, which are 1, 2, 3, and 4. On the left-hand side, we have the cosmic database. On the right-hand side, we have a more up-to-date database, and they still fall into the same signature groups, which is good to see that our signature groups can be classified similarly to what other signature groups have previously done. So there's nothing unusual about our tumor profiles. What we were able to do with this is to actually identify what are the major factors associated with tumor development in these patients. And it turns out that it's related to DNA repair. As I said earlier, I was going to talk a little bit about this. And it's basic excision repair. If you get deamination, spontaneous deamination of a 5-methylcytosine, that needs to be repaired by a DNA repair process. And most of these end up changing a cytosine to thymine, and this can have consequences with respect to whether or not a tumor suppressor gene may be shut down or shut off, or indeed it may activate a protein. On the left-hand side, again, this is the cosmic database, and on the right-hand side with further information from additional tumors from around the world, and as we contribute to this, these matches indicate that DNA repair, especially mismatch repair, which incorporates basic excision repair, is an important factor associated with tumor development. So from the signatures that we have identified, we can state that a significant number of tumors are linked into the amination of 5-methylcytosine, and that is repaired by a DNA mismatch repair, basic excision repair pathway. Of note, the number of variants are rare in constitutional DNA, but common in tumors. An inverse relationship between the number of copy number variants and tumor grade appears to be associated with survival in our brain tumor cohort. Some somatic changes in individual genes are associated with survival. Somatic gene trios are also linked to survival, as I've indicated. The two mutation signatures appear to be particularly important. One is the deamination of 5-methylcytosine, and the other one is DNA mismatch repair. And finally, the evidence from what we presented here today suggests that epigenetic studies will provide more definitive relationships to be identified within this cohort and potentially other cohorts, and it's something that we're currently underway with. And with that, I'd like to end the talk and open it up for any questions. Thank you very much.
Thank you so much, Professor Rodney Scott, for a very detailed presentation. We will be taking question and answer at the end of this session. So I will now request Professor Tyler Dog, Professor of Gynae and Ops Research, who came all over from Hanover Medical School. Thank you, sir, so much for coming for his talk on genomic research risk factor for cervical cancer. Professor Tyler Dog.
Good afternoon, and thank you to the organizers for inviting me here to Peshawar, in particular Professor Muhammad Usman Rashid. It's a pleasure for me to be here and to share with you the latest insights about genomic risk factors for cervical cancer. When we think about cervical cancer, we probably would not think of genetics first, because we all know that cervical cancer is a virally driven tumor. It is driven by human papillomavirus, and there is a very effective vaccination in place. But on the other hand, we also know that HPV infection is not sufficient for progression towards cervical cancer. Some 90% of infected women will not progress into invasive disease, and the reasons for that are poorly understood. There are certainly some epidemiological risk factors, such as smoking and others, but they are also not fully explaining these differences between women who progress and those who don't. We know that there is an increased familial relative risk of about 1.8 for those who have a mother with cervical cancer or sister with cervical cancer, although we don't see very many multiple case families. So the familial relative risk is significant, but it's not sufficient to give rise to multiple case families in large numbers. But nevertheless, this suggests a genetic predisposition, and this has been the topic of some research for over 25 years now. We don't know very many genetic modulators for cervical cancer, in contrast with breast cancer, ovarian cancer, endometrial cancer. This is a relatively under-researched field. But if you look from the left to the right at the progression towards invasive cervical cancer, there are many steps. There are host factors could contribute to a genetic modulation of the disease. We have the modulation of immune response, because it is a virally induced cancer. The virus integrates into the genome, so we also can think of modulation of viral integration. Then the virus expresses oncoproteins, which drive the transformation of the cell. So we could think of modulation of oncogenic transformation by interaction of host factors with the viral proteins. And then the genome instability drives the further evolution of the tumor, and that also may be prone to host factor modulation. When we look at other common cancers, such as breast or ovarian cancer, then we have learned from those cancers that there are different groups of germline variants. We have, on the one side, high-risk variants, which are fortunately rare. On the other side, we also have low-risk variants, polymorphisms, that are common but only confer a small increase in risk. And we have the intermediate variants, which are relatively rare and have a relative risk of 2 to 3-fold. And these variants, or these types of variants, can be captured by next-generation sequencing, in particular suitable for identifying high-risk rare variants. Or if you look for common polymorphisms, then it may be possible to identify them by large-scale comparison of cases and controls in genome-wide association studies. And both of these approaches we have taken for cervical cancer at Hannover Medical School. So I will first talk about our next-generation sequencing results, and we have employed 1,500 samples from patients with invasive cervical cancer, 1,500 patients with cervical dysplasia, and 1,500 healthy females. And these samples have been collected by the German Cervigen Consortium, which we have started some 12 years ago. And you see on the left side the 9 participating clinics all across Germany. What we have done by now is gene panel sequencing, and we first have looked at 12 genes that are involved in homology-directed repair or in mismatch repair. So 7 genes that are known as cancer genes in breast cancer and ovarian cancer, for example, and 5 genes that are known for mismatch repair syndromes such as endometrial cancer. And we have taken 524 German cervical carcinoma patients, and you see that there is a slight distortion from the normal distribution of histiotypes. So we have taken all our adenocarcinomas, which usually only constitute 20% of the series, and compared the 144 adenocarcinomas with a similar number of squamous cervical carcinomas. And we also took 216 dysplasias of high grade. And we also took all 99 HPV negative cervical carcinoma patients, or perhaps I should say HPV undetected, and compared them with the 321 HPV positive cases and 104 with unknown or undetermined HPV status. And this series of 524 German cervical carcinoma patients was then subjected to highly parallel multiplex PCR and Illumina next-generation sequencing. And then we sorted out the pathogenic variants, which we define as truncating variants or splicing variants. And missense variants are a bit difficult to classify, as you have heard in the previous talk by Professor Rodney Scott. And we have decided to take the most recent artificial intelligence-driven algorithms to predict missense variants, that is ESM1B and alpha missense. And only if both these tools predict pathogenicity, then these missense variants were classified as pathogenic. All the pathogenic variants were then subjected to Sanger sequencing validation, and then we looked at the clinical parameters of the carriers. So we identified 29 carriers, which is around 5%, with pathogenic variants in HDR genes, the homology-directed repair genes. And these include 2 in BRCA1, 5 in BRCA2, and 2 in PALB2, which are known breast cancer susceptibility genes. and also 4 in BRIP1, which is a known ovarian cancer susceptibility gene. 13 of those were in squamous cervical carcinoma and 8 in adenocarcinoma, so it's sort of equal distribution, and 8 in dysplasia. And 19 were in HPV positive cancers, only 5 in HPV negative cancers, perhaps contrary to what we hoped to find. But we have no evidence that the BRCA1 or BRCA2 or PALB2 carriers drive HPV negative cervical cancer. We also identified 11 carriers of pathogenic variants in mismatch repair genes, and these include 5 carriers in Lynch syndrome genes, MLH1, MSH2, MSH6, and PMS2, and 6 carriers of the low-risk mutations in MUT-YH. Interestingly, in this case, almost the large majority was found in adenocarcinoma. And 6 of these were in HPV positive cancers and 5 in those where HPV status had not been determined. So we do not have evidence that mismatch repair mutations drive HPV negative cervical cancer. But there is a trend towards an enrichment in adenocarcinoma, which is borderline significant, not yet significant, but numbers were small. So to sum up, some 2 to 3% of cervical cancer patients in our series harbor known predispositions for hereditary breast and ovarian cancer. That is about 4-fold more than expected by chance. And some 1% of cervical cancer patients genetically indeed have Lynch syndrome. The preliminary observations that we should follow in future studies is the enrichment of MMR gene variants in adenocarcinoma. And we also see that MMR gene variants were associated with HPV positive status. And that is an argument against a misclassification, because there is a discussion going on that cervical cancers with microsatellite instability may be misclassified adenocarcinomas of the lower uterine segment, but adenocarcinomas of the endometrium usually are HPV negative, and we don't see this here. We also looked at the age at diagnosis, and the age at diagnosis for the invasive cervical cancer cases was lower in the HDR mutation carriers and also in the MMR gene mutation carriers, but this was only slightly lower and was non-significant. So these results that are presented could be interpreted in a way that there is some slight increase of mutation carriers for the known cancer risk genes in cervical cancer, but they by far would not explain the heritability and the increased familial risk in cervical cancer. So there must be more than we tested in this first part. And now I come to the second part and would like to discuss the genome-wide association studies. And this is based on the polygenic risk model. In the polygenic risk model, we assume that these two groups of cases and controls are differing by the number of low penetrance polymorphisms. And while every low penetrance variant is common and contributes only a very small risk, the accumulation of these low risk variants may lead to a substantial risk of 2 to 3-fold. And the first genome-wide association studies that addressed this question by analyzing all known polymorphisms in hundreds of cases and controls date 10 years back, and they came from a Swedish group who was the first to analyze and investigate or find out that HLA variants are associated with cervical cancer. And another one comes from a Chinese group and identified loci at chromosome 4 at XOC1 and 17 at gasdermin B. But for some time, there was no further genome-wide association study. The Swedish group corroborated the association with the HLA region in a number of replication studies. But it took until the year of 2019 that we then saw the next genome-wide association studies. We have 3 small genome-wide association studies, including one from our group, and 2 large association studies based on national biobanks, in particular the UK Biobank and the Finnish Biobank contributed here very much. But as it is always in science, the one study does not necessarily confirm the results from the other study. And there are hits per study which were not found in the genome-wide association studies from other groups. So it's still necessary to replicate those findings and to fix those variants which definitively are associated with an increased cervical cancer risk. And that is where our Cervigen study entered first. So we aimed to replicate these GWAS loci, and we were successful to replicate a number of variants at the HLA locus in chromosome 6. There are 8 associations, but the HLA region has a substantial linkage disequilibrium. It's a critical region with many genes and many linkage patterns. So when we did conditional logistic regression analysis on these results, we found that there are 3 independent signals, and these signals are close to the genes HLA-DRB1, MIC-A, and PPX2. This is published data in the International Journal of Cancer. 2 of the 3 lead variants are associated with long-range regulation of several HLA genes, which means that it's not necessarily the adjacent gene to that GWAS signal that is really the gene of interest. There are many genes coordinately regulated by these variants. A second locus was then found at chromosome 2, and this locus encodes the transcription factor PAX8, that is where a variant is significantly associated with cervical cancer and dysplasias, and is also associated with PAX8 levels in HPV-positive tissues. And a third locus that we could replicate encodes Gas-dermin B. There are 3 reported signals from 3 different GWAS studies, and we think that is all just can be traced back to one particular splicing variant, which is the likely causal variant. And this variant leads to exon 6 skipping in Gas-dermin B transcripts, and that blocks pyroptosis. Pyroptosis is an inflammation-associated form of cell death, which could be important in the development of dysplasias. We then went on and asked whether there could also be previously overlooked signals because they are HPV type specific. As you know, HPV has several pathogenic types. So the vaccination, for example, is nonavalent, includes HPV 16, 18, 33, 45, and so on. And the question is whether some risk variants for cervical cancer may modulate HPV infection for a particular type or the progression for a particular type, and then should be pre-identifiable using an HPV seropositive GWAS. And the HPV seropositivity GWAS can be done on healthy females. So you have many more females in this GWAS. The power is high to detect HPV seropositivity. And there is one study from John Wittes' lab in genome medicine in 2020 from Stanford. And they indeed investigated the landscape of host genetic factors involved in immune response to common viral infections. And they came up with an HPV 16 specific signal and an HPV 18 specific signal, which we then took for replication genotyping in our Cervigen series to assess the risk of cervical cancer. And as you may see from the table 1, we indeed succeeded to confirm the HPV 16 seropositivity signal in HPV 16 positive invasive cervical cancer and the HPV 18 seropositive signal in HPV 18 positive adenocarcinoma. So this is sort of confirmatory evidence that we can use such a seropositivity GWAS for identifying additional risk factors, and there may be HPV type specific signals. So in the rest of my talk, I would briefly touch the Cervigen GWAS that we have done. We have subjected 375 cancer samples and 866 healthy controls to the OncoArray that directly genotypes 600,000 variants. And after imputation, we had results for 21 million variants. And we didn't find a genome-wide significant signal, but we identified 3 loci where the p-value is less than 5 × 10⁻⁷. And we did subsequent replication genotyping in the whole Cervigen series, which confirmed one association, namely the 14q association. We functionally followed up the signal with luciferase assays, which indicates that there is indeed allele-specific regulation of gene expression if such a haplotype is present. And the EQTL analysis in cervical tissue suggested FOXG1 and 2 link RNAs as likely targets. We don't know much about the 2 link RNAs, but the predicted targets include onco genes such as P16, which is important for cervical cancer. So we think that we have 2 promising link RNAs here as potential risk factors for cervical cancer. A postdoc in my lab also did the genome-wide gene analysis using MAGMA with sliding windows of 25 kb around genes, and the cumulative association then of all the variants in that window was investigated for 19,000 protein coding genes. And you see that one gene stands out, namely OSGen1. And this is a gene encoding oxidative stress-induced growth inhibitor 1, a p53 regulated DNA damage responsive protein, which is under investigation in my lab. So to summarize, we don't see a clear role for classical HDR or MMR genes in cervical cancer etiology, but we do see some enrichment of MMR variants in adenocarcinoma, which deserves further investigation. We have now a number of GWAS identified risk factors confirmed, including HLA locus and PAX8 and gasdermin B. We also have seen that there are HPV type specific risk factors, and our own GWAS identifies a region on 14q12 and a novel candidate gene, which we need to follow on further. And the reason why we do this, of course, is that we want to understand the disease pathways better and eventually develop polygenic risk scores for future risk prediction. My time is over, but the most important slide is, of course, the slide acknowledging these people involved. There are many people involved, but probably the most important one is Danya Ramachandran, who worked as a PhD student and then as a postdoc here in my lab, and she's the driving force behind all our cervical cancer studies. And I also would like to acknowledge the German Cervigen Consortium and the very nice collaboration with the University of Cambridge, a long-standing collaboration with Doug Easton, who helped to provide the OncoArray for the GWAS analysis. And our work was funded by a charity foundation, namely the Bruno and Helene Göster Foundation in Germany. Thank you for your attention.
Thank you, Professor Thiele. So our next speaker is Dr. Thomas Kistlinger. He's a senior scientist, University of Toronto, Canada. So he's going to talk about the usage of quantitative proteomics for the early detection of colorectal cancer. That would be a virtual talk.
So thank you for inviting me to this exciting meeting. And what I decided to show you today is a mix of published and unpublished data where we combined patient-derived xenografts with glycoproteomics for the identification of either liquid biopsy, serum biomarkers, or surface therapeutic targets. So this is how I set up the talk. First part is, as I said, published. So we use patient-derived xenografts and glycoproteomics in the context of high-grade serous ovarian cancer for the discovery of novel serum biomarkers. And then we use this data to systematically develop targeted proteomics assays, so-called parallel reaction monitoring, or PRM assays, basically like a mass spec version of an ELISA assay. And we applied these assays to the evaluation of these biomarkers in longitudinal human serum samples. I will show you then some unpublished data, how we further validated these markers in additional cohorts. And at the end, I'll show you a bit of a sneak peek of how we extended this PDX strategy to 7 additional tumor subtypes. So, and these parts are unpublished. So first part deals with ovarian cancer, so a little bit of background. So epithelial ovarian cancer is the most lethal gynecologic malignancy in Canada, and is usually diagnosed at an advanced stage. And these ovarian cancers can be subdivided in 5 histological subtypes that you can see here in this bar chart on the right-hand side. And they can further be each of these subtypes can further be subdivided into many molecular subtypes. And that's based on mainly profiling studies like the TCGA. High-grade serous ovarian cancer is the most common and the most aggressive subtype, and the subtype that we are interested in the context of this project. The vast majority of high-grade serous ovarian cancer patients present with advanced disease, so where this cancer spread throughout the entire abdominal cavity at presentation. And so that suggests early on this disease is silent and is hard to detect. Early detection is very difficult. Also has a very interesting cell of origin. So most of these cancers either originate from the ovarian surface epithelium or from the fallopian tube epithelium. And then very early in the progression of this disease, or in the development of this disease, cancer spreads from these primary sites throughout the abdominal cavity, and often to fatty tissues such as the omentum. So at the end of the day, at presentation, most of these cancers can be considered a metastasis. And here you can see what this looks at presentation. So there's massive tumor load throughout the entire abdominal cavity, often with liters of acidic fluid as well. Treatment has not changed much in the last decades, so usually includes aggressive debulking surgery followed by chemotherapy. And these chemotherapies in first line often combine carboplatin and Taxol. And then following treatment or following surgery, CA-125 serum levels, so this is a FDA-approved glycoprotein biomarker, are used to evaluate efficacy and recurrence in this disease, efficacy of treatment and recurrence. And there's a little bit debated if this has any effect on survival of these patients, with many, many commentaries and reviews written on this topic. Most of these patients recur within 2 years. This is most likely due to incomplete debulking. And as a result, 5-year survival is low, 10 to 30%. And here on the right-hand side, you can see a typical longitudinal profile of CA-125 in an ovarian cancer patient. Starts often high at diagnosis, drops through surgery and chemotherapy into remission, usually goes below 35 units per milliliter, and then rises again in recurrence. Often these patients are then resistant to first-line therapy, get second-line therapy, often develop resistance there as well, and eventually succumb to this disease. 20% of patients do not have detectable CA-125 levels, so they actually at the moment do not have any biomarkers that can be used to evaluate efficacy of treatment or measure recurrence. So we are interested in defining new biomarkers that have similar longitudinal profiles and of tumor load, as we call this here. And we're particularly interested in this phase of disease progression. So ideally, potentially a marker that can detect minimal residual disease better or recurrence earlier than CA-125, which rises usually 4 months prior to clinical symptoms. So here's the project that a former student, Ankit Sinha, designed in the lab. So we utilized patient-derived xenograft models developed by the Neil and Ailes lab at Princess Margaret Cancer Center, but we reestablished them through IP engraftment which has slightly different characteristics than was developed here in this PNAS paper through mammary fat engraftment. So at end stage, we surgically remove tumor tissues, collect serum from these engrafted animals, and collect serum from unengrafted animals as negative control. And these are immunocompromised NSG animals. And then we have this kind of complicated glycoproteomics / bioinformatics workflow. So here's kind of, you see the chemistry of this strategy. So we lyse our proteins, we spike in a yeast glycoprotein invertase, gene name SAC2, that allows us to normalize in between samples. We digest proteins to peptides, and at this stage, we're oxidizing glycans with metaperiodate to aldehydes. So cis-diols to aldehydes. And then we use magnetic hydrazide beads to covalently attach to these formed aldehydes, forming a hydrazone. That's a stable covalent bond that allows us to rigorously wash these samples, remove the vast majority of non-glycosylated peptides, and then we release these peptides from the beads to an enzyme, PNGSF, that cuts between the asparagine, where that sugar is attached to, and the first sugar residue, converting that asparagine to an aspartic acid. That's a 1 Dalton mass shift within a specific consensus sequence that allows the mass spec to detect this modification introduced by the enzyme. We can then run these samples by mass spectrometry, use, as I show you in the next couple slides, species assignment. Is this peptide derived from the host mouse or from human, the engrafted tumor? And then use this data to select candidates for the development of targeted proteomics assays, these PRM assays. And why are we interested in glycoproteins? So glycoproteins are classically secreted and cell surface expressed. So that's what that co-translational modification is for. It targets proteins to these localizations. These proteins are particularly interested in the context of fluid biomarkers. These glycans also provide stability to that protein against extracellular proteases in the blood. And as a result of that, most FDA-approved blood cancer biomarkers are glycoproteins, such as C125 or PSA. So here you can see, after running all these, this is what we obtained. Detected around 4,000 glycopeptides from 2,000 or so glycoproteins. And this is how they're distributed among these 3 sample types. And then using subcellular localization annotation, you can see that the vast majority, 98%, of these glycosides that we detect are from either secreted or plasma membrane proteins, particularly in the ectodomain. So that's to be expected. It's a nice control. Okay, so here's one little feature or trick of these patient-derived xenografts that we utilize here. So a patient-derived xenograft tumor is basically a mixed species tissue where it's human from that patient-engrafted tumor, and the stroma is from mouse. And we can bioinformatically dissect where these peptides came from human or from mouse. And this is kind of indicated here in these charts on the right-hand side. So let's focus on the first one here. This is 1,600 or so glycopeptides detected in the serum samples, sorted by intensity or abundance. So the most abundant here, the least abundant here that we detect. And on the bottom in the stacked bar chart, we split them in abundance septiles. So here, for example, the 230 most abundant, the 230 least abundant. And then within these septiles, we assign species to these peptides. In red, it's a human unique peptide. In blue, a mouse unique. In green, that species is conserved between the two species. And you can see as the abundance decreases, the percentage of human-derived peptide increases, and that's to be expected. Proteins that are secreted from that tumor into blood will be a minority of the blood proteome background, hence they should be less abundant. Conversely, in the tumor tissue, there's no such difference, and that's also to be expected. There's nothing that suggests that a human or mouse tissue or proteins from human or mouse tissue would have any kind of bias in that tumor. they should be equally distributed throughout the abundance range. Okay, and then I'll summarize this data in a couple of cartoons before I show you some results. So now we use this data to specifically select peptides for which we were interested in generating PRM assays. And we, of course, were most interested in these peptides that overlap between the engrafted serum samples and the tumor. We're probably a little bit overambitious and also took all the peptides that were tumor unique as well. We then go through a number of mining strategies. We want these peptides to have certain biophysical properties that allows us to detect them properly by mass spec. And we also account for certain post-translational modifications that are introduced as a result of digestion and handling. I'll make a long story short, there's around 450 peptides that we selected that are human-derived, detected either in serum or in tumor, that we built now these targeted proteomics assays. So imagine it's like conducting 450 ELISAs in a 2-hour mass spec run all at the same time. So there's a huge number of detections and quantitations that are happening here. And then we applied these assays to 2 independent longitudinal human serum samples from high-grade serous ovarian cancer patients collected at different times through disease progression. So here you can see it, 450 assays for which we developed these proteomics assays. And then we have 2 different cohorts, 10 patients initially with 50 samples because 5 time points. And all of these serum samples have matching FDA-approved serum ELISA data for CA-125. And this is what CA-125 ELISA looks like longitudinally in these patients. High at diagnosis, goes down through treatment, stays low in remission, rises again in recurrence. So we applied the 450 assays to the first cohort and then mined them very rigorously. Reproducible detection, mimicking these tumor load profiles. But we were particularly interested in proteins that actually rise early, that rise already during remission and then further rise during recurrence. 27 showed reproducibly these longitudinal expression profiles. We optimized these assays and applied them to the second cohort. 4 still showed these expression profiles. And here you can see just some data. So this is the actual 50 serum samples from the 10 patients with the serum ELISA values. You can see high comes down and then rises again. And then here's our proteomics data in that same cohort, normalized to the diagnosis time point. And you can see they come down after surgery and chemo, but start to rise already in remission and then continue to rise as these patients recur, defined by rising of CA-125, usually over 35, or by some imaging type clinical assays. Okay, so that's interesting. Just to summarize this is part of this first published paper here. So we could show that PDX models in combination with glycoproteomics and shotgun proteomics allows you to detect putative human biomarkers that you can then develop systematically targeted proteomics assays and verify them in independent, longitudinally collected human serum samples. Of course, so what's the next step? First step would be, can we further validate them? Now we only have 4 markers. Now you can really start optimizing these proteomics assays and increase their sensitivity. That was done by a postdoc that we went in the lab, who's a fantastic analytical chemist. So it was a tailor-made project for her. And then at the end, I'll show you also how we utilize this PDX strategy in a large scale, and that's done by a PhD student, Minusha Govindarajan. I'm applying the strategy to 7 different tumor types. So here, first validation. So now we have a slightly larger cohort, 34 unique ovarian cancer patients, 212 samples collected. So on average, around 7-ish samples per patient over time. All have matching CA-125 ELISA data. And then here's the same protocol that I described to you earlier, but we tweaked certain things. So for example, we went from 50 to 25 microliters. We optimized capture of these glycopeptides. And then most importantly, we optimized the mass spec, the LCMS conditions. So we reduced chromatography time to 45 minutes, So that allows you to really get some throughput now. And then we dramatically optimize mass spec setting to improve sensitivity of these assays. And then apply them again to these patients looking for these tumor load longitudinal profiles that are indicated here in this cartoon. So here, 2 years of work summarized in 2 kind of heat maps. So we mined the data in 2 stages. And I really here in this figure, what's important is this up here and the heat maps down here. So first part, we ask, do markers mimic the reduction in abundance as a function of treatment? So following surgery and chemotherapy, do the markers drop in abundance? So here you have the 4 PRM assay compared to the CA-125 ELISA, where blue indicates that over time, these proteins drop in abundance. Red means they're rising. So that shows you that all these markers, not in every patient, do actually go down as a function of treatment. CA-125 ELISA, optimized for 25 years, still performs the best here. And then alternatively, do it after the end of treatment in remission and recurrence, do these markers rise again? So here again, red means rising, blue means go slightly down, and gray means nothing happens. So again, CA-125 does quite well, and all these markers on average in many patients all do rise as a function of recurrence. And then I'm not showing you, there's all kinds of technical aspects that go in here, but LTBP1 is a very interesting marker that technically performs extremely reproducible and robust. So specifically focused on that, and we took a further look. So we asked now a harder question. So a subset of these patients, we have multiple remission time points. And we asked the question, is there indications, as we had in the original paper, that LTBP1 rises potentially earlier than CA-125 in recurrence? And you can see this here in these charts. So again, normalized to the diagnosis time point. So you can see in pink, CA-125 drops down, stays down, and maybe at the very end starts to rise until it hits recurrence. The signal to noise of this assay at the moment for LTBP1 maybe not as high, and may even have a little bump after surgery. Nevertheless, goes down following surgery and chemotherapy, but then in remission, it seems to be rising earlier than CA-125. That's statistically significant, by the way here. And you can see it maybe in one given patient here pulled out. So you can see these longitudinal quantification tracks. CA-125 comes down, stays low, and really only in recurrence kind of rises. And CA-120 and LTBP1 seems to be rising earlier. So this is, of course, not absolutely validated yet or clinically implementable, but this at least suggests that this is a marker that we can now dig our teeth in and really figure out, optimize assays further. So it certainly is a marker that mimics tumor load profiles, but we're interested now, can we further confirm that this indicates recurrence earlier than CA-125, which could give these patients alternative treatment strategies in that short window they have before they get on in the second line treatment window. And then just, I'm good in time. So here I'll show you a little bit of a sneak peek of our PDX-based strategy. So now we expanded from high-grade serous ovarian cancer to 6 additional tumor types: lung adeno, head and neck, GBM, lung squamous, pancreatic, and colorectal, performing the same strategy. So it's 85 unique PDX models. For some of these, we also have matching primary cells or immortalized cell lines that are specifically selected for a tumor type. And we perform a similar strategy, but mapping proteins at the cell surface. And here now we're interested in looking at tumor tissue detecting surface antigens, so potential therapeutic targets for the development of antibody-based immunotherapies, such as antibody-drug conjugates or CAR T cells. And we mined this data in a certain way. So we have about 1,300 human unique proteins, about 700 are robustly detectable in each of these tumor types. Many of them have limited expression in normal tissues, so we use public data to evaluate this. You want as little as possible detection in normal tissue to minimize off targets. And then we benchmark this against 30 clinically approved or in phase 2 trials, ADC or CAR T targets. And we use that as a benchmark to estimate, do our markers look better in the context of abundance and expression in normal tissue, or at least equal to these markers. And you can see how we kind of mine this, right? So we use a three-pronged mining strategy. So we take these 1,300 proteins and ask, okay, are they abundantly detected and at least detected in 50% of the PDX models in our data? Are they glycoproteins that are also predicted to be surface targets using this prediction algorithm? And do they have minimal expression in either the Human Protein Atlas or GTEx RNA or proteomics data? And we use a kind of a rank scheme to come up with a score that compares them or benchmarks our candidates against these ADC and CAR T targets that we have co-detected in our data sets. You can find for each tumor type in the low hundreds surface markers that are unique to these tumor types. And there's many of the current ADC or CAR T targets expressed in them, but also emerging ones. So GBM, for example, one of our top target is CD97, and just a paper was published in bioRxiv that shows that CD7 is a promising ADC target in GBM, giving further proof to this kind of a strategy that we developed. And of course, now we're interested in studying function of many of these molecules in the cell culture model using kind of CRISPR strategies in the future. So here, to summarize this data, the second part of the talk, we optimized our PRM assays, applied them to an independent clinical cohort, and have identified LTBP1 as a potentially novel tumor load biomarker detectable in serum of high-grade serous ovarian cancer patients that potentially detects recurrence earlier than CA125. We have expanded our PDX platform to 7 additional tumors and are interested now in utilizing this data for the development of immunotherapies against tumor surface antigens, but also to study interesting tumor surface marker using classic molecular biology. And with this, I would like to thank the group that really did the work, clinical collaborators, and you for listening, and happy to take any questions in the live session. Thank you.
This is Dr. Thomas for a very informative talk and very innovative work. So the last presentation for this session before we go into the panel discussion is Dr. Li Wang, in which EDU axis regulate immunoimbalance in the tumor microenvironment. He was in Newcastle University, UK.
Hello, everyone. My name is Lei Huang. I'm a senior lecturer in Newcastle University. Thanks for the host to invite me into this wonderful symposium. Unfortunately, I can't be here in person. So hopefully you'll enjoy this talk. My topic of my talk is the STING ideal axis regulates immune balance in the tumor microenvironment. So I want to start with the question on how do some people get cancer? And cancers are recognized originally for a long time as cell-intrinsic diseases. And cancer cells acquire mutations. The cell acquire mutations, genes controlling division and survival, or they can be changed epigenetically in controlling cell division and survival, or they can be experienced with oncogenic infections carrying oncogenes from viral origin. And those cells lose growth control and survive to form pre-malignant lesions. And after long time development, they start to form invasive tumors over time. And the traditional therapy includes kill or remove cancer cells, and that's the main focus by surgery, chemo, radiotherapy, and more recently, the newly targeted therapy was developed using tyrosine kinase receptors to block the growth hormone or to block those growth factor interactions to inhibit cancer cell growth, which has been quite effective, but it's hard to eradicate cancer. But is that all? And is that all we can do with cancer? Certainly not. In the past 10 years, there are new perspectives of cancer recognized. Now, cancer is, we think, is chronic inflammatory diseases. The main reason for that is most of immune cells, most of the cancer develop very early stage and becomes the cell mutation develop early, but become cancer much later in the life. And this is because immune cells prevent pre-malignant lesions to form tumors or malignant cancers. And that procedure is called immune surveillance. And occasionally, inflammatory cells will create microenvironments which are conducive to tumor growth. And that procedure is called immune escape or cancer immune editing. And at a clinical presentation, most of patients has a very mature tumor microenvironments, which including many factors, including cancer-associated fibroblasts, tumor-associated macrophages, and etc., and some protein factors called immune checkpoints, which can be used for therapeutic targets, etc. And those are immune suppressive and to suppress natural and vaccine-induced immunity against tumor antigens. So some therapy reinforce suppressive tumor microenvironment despite being effective in controlling tumor. And these are typically traditional therapy, especially for radio and chemotherapy. So based on these, the new therapy strategy has been developed by killing cancer cells using immune cells and disrupt the tolerogenic tumor microenvironments. And those are called immunotherapies. And just to go a bit further on immune checkpoints, because these are now being widely used in clinic already, especially for the first two, CTLA-4 and the PD-L1. And antibodies have been developed targeting these, what we call immune checkpoint blockade therapy. And these agents, these suppressive immune checkpoints, ICPs, they actually mediate some DC cells to T cell interaction to cause T cell to be suppressed, and as well as DC T cell interaction and with Treg as well, and that causes T cell to be functionally suppressed. And these two persons, James Allison and Hongzhou, was awarded a Nobel Prize because of the discovery of these checkpoints. One more checkpoint is being discovered early 2000 was by Professor Andrew Mellor in America, Georgia. It's called indolin 2,3 tryptophan catabolism pathways, including three enzymes called IDO1, IDO2, and tryptophan 2,3 dioxygenase, TDO. And it was originally discovered as a maternal immune tolerance mechanism in placenta. So in this case, if the father and the mother are have identical genetics, then the baby will have identical genetics with the mother. So in that case, if inhibiting IDO, there's no fetus rejection. But if the father and mother have different genetics, so the baby will carry half of genetics from father, and mother will recognize those as a foreign antigen, and they will mount immune response against it. In that case, but inhibiting IDO causes allogenic embryo rejection, cause these bleeding in the placenta, in the decidual areas. And that's a seminal discovery at the time, eventually being used into other areas like autoimmune disease and transplant, as well as the tumor biology as well. A little further on immuno-Rx, so immunotherapies. There are four classes of immunotherapy can be mainly recognized. ICB, including agents targeting PD-1, CTLA-4, and IDO, which IDO is one of the major topics I will introduce today. And second is called adjuvant therapy. Those are the reagents will incite inflammation when you inject into tumor. They cause inflammation. And one of those typical examples is the STING agonist by targeting STING. And the third is the vaccination. So you can use vaccine to induce anti-tumor immune cells to be effectively working the tumors to eradicate tumor cells. And the fourth is called adoptive T cell transfer therapy by transfer from a third party or self-cultured immune cells recognizing tumor antigen back into the host and then cause elimination of tumor. So these therapy, a lot of them are quite promising from clinical results, also the preclinical data, but yet much to be learned. It's because so far, the best therapy like PD-1, in the best day for most of cancers have about 20 to 50 response rates. And 50 is only for melanoma. There's no other cancer types for that. Things like head and neck cancer, almost no patient responded to that. And so this is a mystery that why some patients don't respond, and why some patients respond to immunotherapy. And we call those don't respond as non-immunogenic tumors. Furthermore, the adverse effects is also quite common, including autoimmunity and neurological complications. And in addition to the immunotherapy, some of the conventional therapy also have immune regulatory effect or immune activation effects. For example, the radiotherapy now is very well documented to activate STING by releasing intracellular DNA. And then through STING, it's actually activated into tumor immune functions. And part of the radiotherapy effects is via its immune activation. So IDO is one of a major metabolic immune checkpoint, and it's induced by inflammation to suppress immunity and commonly in the pathogen infection as well as the tumors. IDO inhibitors have been widely tested to boost anti-tumor immunity in animal models as well as in clinical trials. The IDO inducers in animal models especially was very well documented to suppress T-cell immunity and to protect healthy tissue as well as the transplant tissue from immune-mediated destructions. The expression of IDO is not only cause immunosuppression, but also leads to chronic neuropathy, like neuropathic pain. And the IDO inhibitors, including indoximod, NLG919, and the BMS drug, especially in 2015, the IP exchange deals reached 6 billion, which became a very hot topic at the time. Of course, there's still challenging appearances exist, remaining to be whether the IDO can be used as a biomarker for stratified medicine, or if we need a better IDO inhibitors to improve the therapeutic functions. So IDO is not only immune checkpoints, it's a hallmark of inflammation, because essentially IDO is induced in all inflammatory conditions. Because inflammation usually associated with the production of prostaglandins and interferons, and IDO is strongly induced by these two factors altogether. So the function of IDO is leading to tryptophan metabolism to kynurenine pathways instead of serotonin pathways. So without IDO expression in most of tissues, tryptophan regularly degrades or metabolizes through the serotonin pathway to form melatonin, which maintain a healthy status. When IDO is induced, not only degrades serotonin to reduce the amount of serotonin and melatonin, but also reduce the substrates of tryptophan and produce these metabolites, including kynurenine, kynuric acid, and 3-HOA, and the quinolinic acid. And these have multiple physiological functions, including immunosuppressive function, as well as neurological functions. For example, QA and the KA2 together, they can act in an opposite way to the NMDA receptor, cause fatigue and pain, as well as depression. On the immune regulation side, the ideal one quickly degrades tryptophan into kynurenine by consuming the oxygen radicals. By depleting tryptophan, it can activate an integrated cellular stress pathway mediated by GCN2, and that GCN2 activation causes T cell apoptosis, Treg activation, and DC regulation developments. But also these metabolites, as I mentioned in the last slide, the cannurin as well as the 3-HAA, and they have a strong function to activate AHR, aryl hydrocarbon receptor, as well as the PDK1 and the MOR receptor maybe. And that causes T cell regulation to suppress the T cell function and to activate Treg, also further suppress T cell function. And the activation of these two pathways altogether, that will give the sites of activation something called acquired immune privilege, meaning this site will be exempted from immune cell attack. And commonly, this is good for the normal tissue, but this is bad in case something happened like a tumor or chronic infection. And of course, some of these metabolites will further cause neurological and behavior changes, including sickness-induced depression and chronic neuropathic pain. So in collaboration with clinicians while we were in USA, we have studied a cohort of we have followed up a cohort of patients for 10 years survival on their these stage 3 and 4 lung cancer patients treated by conventional radiotherapy. And we can, using the blood ideal activity measured by cannurin, to 4 groups, and then including the pre-therapy low, post-therapy low, pre-therapy high, post-therapy low, and pre-therapy low, and post-therapy high, and both high. And we can see that in the if no ideal induced at any time of the therapy, and these patients, more than 60% of those lung cancer patients survive. While in the both high groups, ideal is always high, all patients die within 3 years. This suggesting that the ideal impedes the clinical response to radiotherapy, and then the ideal metabolites measuring its activity is a good biomarker of patients responsive, of those patients responsive to RT. So when we start this study, ideal and sting interaction, we actually start from a different angle. So we were trying to use a DNA nanoparticle here to treat autoimmune diseases by introduce ideal gene, this is using a gene transduction period. So we're initially using ideal carrying DNA construct, trying to express DNA, trying to express ideal in the host. Nevertheless, this approach never worked. Well, instead, we found out that these DNA nanoparticles, regardless of what they carry, they actually induce ideal in the host animal. So then we tested whether these DNA nanoparticles actually can suppress autoimmune arthritis via ideal induction. So in this experiment, healthy joints in untreated mice is stained by red color in the joint surface. In arthritic mouse, you see all of these red stainings are gone. So these are proteoglycan staining. That means the joint is damaged. Then in the therapeutic group with DNA nanoparticle, the joint become healthy again. They actually recovered, but in the ideal knockout mouse, and this joint health never recovered, indicated that DNA nanoparticle actually can induce ideal and have a therapeutic effects to protect the joint from an immune attack. And furthermore, 1 more year or 2 more year study, we realized this is by activating a pathway called CGAS sting and cytosolic DNA sensing pathway. So when we have DNA delivered into the cells, it was sensed by a protein called CGAS, and the CGAS, and CGAS will make something called CGUMP, 2,3 CGUMP, and that will bind to sting as a secondary message, as a secondary messenger, and sting activate it, and then to activate the transcriptional complex, including NF-kappa B and IF3, to make inflammatory cytokines and type 1 interferons. And furthermore, these cytokines all together, they will induce IDO and a PD-L1. And that's the general pathway we summarized in the last 10-year study, and was all published using different animal models of autoimmune diseases. And while we start to move to tumor, because we think, whatever produced by whatever protective tissue in these autoimmune diseases may be a factors of protecting tumor when the tumor develops as well. So we want to see whether in the homeostasis condition, whether in tumor microenvironment, the ideal is induced via STING. So what we can see here is we're using a weakly immunogenic model, means these tumor models not responding to any previous known immunotherapy. So in this model, when we inject the tumor cells into the mouse, we see the ideal is induced. And then in the STING knockout with versus interferon type 1 receptor knockout, no ideal is induced. And we can confirm the same observation in the secondary tumor model using another tumor cells called EL4. Furthermore, to observe the tumor growth in these using the genetic modified mouse, so we will observe that if the STING is deficient or ideal is deficient, the tumor grows slower in these mice compared to wild type, suggesting in a wild type mouse the presence of STING and ideal is actually the promoting factor for the tumor to grow faster. And then we further analyzed this by trying to understand whether immune cell play any function in that. And we depleted cytotoxic T cells, which are the T cells killing or controlling the tumor. And after these T cells depletion, we observed that the tumor growth from the STING knockout and ideal knockout is similar, comparable to the wild type, suggesting this effect is mediated by immune function. So secondly, the antigenicity also influences the role of STING. We actually make a new antigen into the LC and just let these cells become more immunogenic by carrying a new antigen. Now we see a shift of the response in the STING knockout versus ideal knockout. So in here, we see in the STING knockout, the tumor grows at similar rates compared to wild type, while the ideal knockout still grows slower, suggesting when the antigenicity change, but then the ideal induction may not be mediated by STING anymore, and STING just lose this tumor promoting effects anymore. While we are busy figuring out how immune suppressive function of STING promote tumor growth, others try to using STING agonist as a therapeutic agents for tumor therapy. And so this is the first report of using a modified stabilized STING agonist in the mouse models, and they show very good effects of therapeutic effects of these agents. And then further along, the clinical trials follows. Nevertheless, later on, it's reported that the compound from Merck have zero activity of anti-tumor activity, even with combined with a very limited response combined with PD-1. While this company, Abdullah, from Abdullah, is relatively safe, but they have really limited amount of 5% partial response to monotherapy. So there's still a need for more understanding and studies evaluating ways to improve the therapy in addition to what they already reported there. So we think maybe what's relevant is that because 80% of the patients cohorts are not responding well to immunotherapy like PD-1, so they are weakly immunogenic. So can we actually change the scenario to make it work? And so now we back to our own model using LLC model. So that's a model not responding to PD-1 or other immunotherapies. So now in this case, when we using IDO inhibitor alone, the third generation ones, the most recent one, it doesn't really have any effects on the tumor growth. But with the STING agonist alone, with CDA, they have a marginal effects reducing tumor growth, but combination have a really nice complete response just after a week of the combination therapy starts. And we further tested 3 different IDO inhibitors to show whether inhibitor are same, and turn out to be they're different too. So with the first generation inhibitor have almost no effects in addition to what's already offered by STING as the black line and the blue line. The second generation have some improvement, while the third generation IDO inhibitor have a very good therapeutic effects up to 30 days, up to 45 days actually at the end of this experiment. But nevertheless, at the end, we see a tumor recurrence and distal tumor in the lung and liver as well. And so maybe could this be just IDO issue? So we further tested whether PD-1 blockade has a similar effects to IDO. And turned out to be, it looks all very similar. So PD-1 alone doesn't have any therapeutic effects. combination have a complete response, but then at the end of the experiments, we see tumor recurrence, and we also see distal tumor in the lung and the lip. And then we test the third agent, cytotoxic, because we know the COX-2 is an important, the prostaglandin is also an important factor to induce IDO, but also at the time we have another angle, we want to see the cancer reduce the pain. Also, we're not going to touch upon here. But what we found is this cytotoxic has a wonderful effects in combination with STING agonist here. So first of all, they completely reduced IDO induction compared to the STING agonist alone. There's no more IDO induced anymore, treated by if the COX-2 is inhibited. Nevertheless, interestingly, the PD-L1 induction by the STING activation is not reduced, suggesting any effects we see here is not related to PD-1, only related to IDO, maybe something else. And then we see a complete response after a week of treatment. But then what's more miracle is that we see all mice were resistant to LLC re-challenge. There's no recurrence, there's no metastasis, but also when we re-inject the tumor cells into the same mouse, no mice had a residue or decidual tumor 60 days after even re-challenge the mice with the same tumor. So furthermore, this therapy also have effects to distal tumor as well. So in this experiment, we actually treated dermal tumor, but while we're treating it, we also inject the tumor cells into the lung by IV injection by injecting the tumor cells systematically. And then we see that only 1 out of 9 mice died. All 8 mice survived with no relapse, no tumor observed anywhere in the body, suggesting the local treatment of the dermal tumor or the local treatment can have a systemic effect to controlling all sites of the tumor in other places. So to summarize what we discussed above, so we think IDO is a good predictive biomarker of clinical response, especially for radiotherapy, at least. And then the role of IDO in the tumor growth is that the DNA sensing to induce IDO, and that regulates immunity in the development. I'm sorry, my mouse is jumping around. That DNA sensing induce IDO and regulates the immunity in the developing tumor microenvironment to promote tumor growth. And then the tumor antigenicity will influence TME and the impact of STING-mediated inflammation. And third, when we use STING agonist as a therapeutic agent, conjugate therapy, the STING is a double-edged sword in treating cancers. So not only induce anti-tumor immunity, but also induce adaptive therapy resistance factors. And all of those immune checkpoints probably are factors induced by that. And the STING agonist can work well, we think, when combined with the right ICB therapy. And this is all my talk. And I want to acknowledge all my collaborators, including Professor Meller, who is my longtime mentor, and my collaborators who did all the lung cancer studies, Professor Spring Kong, now moved to Hong Kong universities, and all the lab staff and the previous mentors in Augusta University, Professor David Mang, who is the pioneer of using IDO inhibitors to treat cancer. Thank you for your attention.
Yeah, thank you, Dr. Li Wang. Very important study and the findings you shared with all of us. So it's now the time for the panel discussion. We have our worthy speakers with us, and if there are any questions, you're more than welcome to discuss it.
Assalamu alaikum, everyone. I am Asim Farooq. I have a question from Dr. Lee. It's about the, like you have mentioned that TDO is also involved in depletion of tryptophan. So if our focus is majorly on inhibiting IDO to control cancer progression and immune editing, shouldn't we also include TDO in this process and simultaneously work on both this? And a second question is, if tryptophan is depleted and we say that the T cells are inhibited because of this, so the tryptophan is also depleted for cancer cells at the same time. So isn't that affecting the growth of cancer cells as well? Thank you.
May I? Yeah, so both are very excellent questions, and we had those all the time. The first question is whether TDL play a role or what kind of role TDL plays. We know that TDL is constitutively expressed in the liver. It's actually an important metabolic pathway to making a critical cofactors for our life, which is niacin or NAD. So the liver always creates such a environment that not favoring the tryptophan. Nevertheless, I think TDL may have a different enzymatic kinetics, so it doesn't really deplete tryptophan to the low level as low as ideal. I think the KM is a bit high if I remember that correctly. So that's one. Second, in the tumor microenvironment, TDL is reported to express in certain tumor types, but most of the cancer type, they don't. They don't really have a prominent TDL existence. And as a matter of fact, that even IDO2 is rarely observed to be functional in the tumor microenvironment. So that's why the IDO1 inhibitor, the ideal inhibitor essentially is actually the most practical way, or most prominent way in treating such kind of disease. I'm not sure that answered our question. And then goes on, second question is the tryptophan depletion, whether cause an effect on the tumor cells. The answer is yes, I would say, because back in 1980s, when the IDO was just discovered by a Japanese group, that was the strategy they have engaged, try to use interferon to induce IDO in the tumor so that can starve tumor to death. The answer is the approach didn't work at the time. And we know that it's tryptophan depletion causes a very specific integrated cellular stress pathway activation via GCN2, while the GCN2 activation has more impact in T cells or immune cells compared to the cancer cells. Cancer cells always have ways to go around. And the other interesting perspective on this is that the tryptophan as an essential amino acids, the cellular level of the free tryptophan is always maintained at a very low level, so easy to deplete. So sometimes I think the cancer cell may also sort of used to the situation, and they can also recycle other proteins to recover by arresting the cell metabolism a bit at the time of depletion. And then they recover very quickly. So this is what we think why this tryptophan depletion did not have a very strong role in cancer cell itself, but rather the effects of immune cells.
Thank you very much. So we have another question for Professor Thomas Kisslinger. The question is, can patient tumor be directly used to optimize the method and skip the animal xenografting and avoid complex data segregation? Professor Thomas.
You could, yes, thanks. I already answered it online, but I'll answer it live too. For sure you could. You could use, I mean, it depends which part of the strategy you talk about. I mean, the first part, the biomarker work, you could for sure use serum or urine or other biofluids as a discovery for soluble biomarkers. Maybe the advantage of the PDX models here is that you can actually unambiguously dissect if that protein in serum was actually derived from tumor or not, from stroma. So that's much harder to do in a serum sample. It's just, I mean, for example, if you're looking for a prostate cancer marker in urine, you ideally have that protein being released from the prostate, not some kind of a bystander effect. That is not so easily doable in patient samples. So we use the PDX as a discovery strategy to earmark possible candidates, but then do go into patient samples to verify them. So absolutely could be done, and we do it as well, but it has unique challenges to it.
Thank you very much. So I have a question from Professor Thilo Dork. So you have mentioned that for HPV specific positive cervical cancer tumors, so what is the clinical spectrum or presentation for women with HPV positive as well as mismatch repair or other genetic variants as compared to those without HPV infection? Thank you.
Yeah, that's an excellent question. So the clinical spectrum is not so much different between those in our cohort. Although there are studies out that claim that there is a different pathology, but we don't see much difference in terms of adenocarcinoma or squamous cervical carcinoma.
Thank you very much. So I have a question from Dr. Lee Hong. So how expensive is this EDO inhibitor? Or can somebody use just celecoxib as a replacement or alternative? Thank you.
From our study of the, do you mean IDO inhibitor or TDO inhibitor? So what I heard is TDO inhibitor. But anyway, the IDO inhibitor at the moment, they are also available from various companies, and especially third generation ones from from BMS. And they are ready to work. They're ready. They're very willing to work with various hospitals if you propose trials and things like that. But you can for non-clinical study, you can also use this from a third-party chemical stores. You can buy these compounds. Nevertheless, from our experience, we found that celecoxib has a very good, very good efficacy to induce ideal induction in general. And we actually test this in various models, and not only in cancer model, but also infection as well. So I certainly, my speculation is the celecoxib would work very well in that, but I don't know anything further, and we just started this not long ago.
I think this was there's no further questions, so it's an excellent session. And this genetic, I by myself, I'm a public health professor. So you're working on the genetic epidemiology and more work on the genes. These biomarkers, they are though expensive, but research like this, what we heard today, that we wish and pray that one day it will be accessible to each and every poor cancer patient, and it's available in a limited cost to the people. For example, people like us, the developing countries. But we, the scientists, working together from different continents and with our hard work and collaboration, this is we have done it in the past, and we will inshallah do it in the future. So thank you so much for taking out time, for sharing your excellent presentations and taking question and answer session. Thank you so much. And I think this is all from our side. Thank you so much.